Okay, so now we will go to the eukaryotic transcription machinery, or the transcription in eukaryotes, or maybe eukaryotic transcription and transcription machinery. So based on the transcription machinery in eukaryotes, the genes, the transcribed genes in eukaryotes, they are categorized in 3 classes. So what do we call them? Eukaryotic transcribed genes. They are categorized into 3 categories. The one is called class 1, another is called class 2, and another is called class 3 genes. So why they are categorized this class 1, class 2, class 3 genes? So the class 1 genes, they are transcribed, they are categorized on this basis that by which RNA polymerase they are transcribed. So the genes which are transcribed by class 1 by RNA pol 1, they are called class 1, and which are transcribed by RNA pol 2, yani RNA polymerase 2, they are called class 2 genes, and those who are transcribed by RNA polymerase 3, they are called class 3 genes. Okay, so essentially the classification of genes in prokaryotes is because of the RNA polymerase they are transcribed with. So basically in eukaryotes, you understand that there are 3 kind of RNA polymerases. So yes, and definitely there are 3 type of RNA polymerases. So this is the type of genes: class 1 gene, class 2 genes, and class 3 genes. Class 1 genes are transcribed by polymerase 1. An example of such genes is ribosomal RNA genes. Ribosomal RNA, exclusively ribosomal RNA genes. We can understand this table in this way, that in the eukaryotes there are 3 kind of RNA polymerases, and what are the features of those RNA polymerases. So the first, the RNA polymerase 1. So we can say the RNA polymerase exclusively transcribe ribosomal RNA genes only, no other genes, only ribosomal RNA genes. Means RNA polymerase 1 transcribe only ribosomal RNA genes. This is one thing. If I say the ribosomal RNA genes are transcribed by only RNA polymerase 1, that's a different statement. RNA polymerase 1 transcribe only ribosomal RNA gene, this is one statement. And if I say the ribosomal RNA genes are, or I say the class 1 genes, okay, so basically class 1 gene, no, ribosomal RNA genes. Ribosomal RNA genes are transcribed only by RNA pol 1, then it will be different statement, and the 2 statements are not identical or not equivalent, because there are some ribosomal RNA genes. For example, this one, 5S ribosomal RNA gene, it is transcribed by the class 3, by the RNA polymerase 3. So this 5S ribosomal RNA is a class 3 gene, it's not a class 1 gene. So the ribosomal RNA genes are both class 1 and class 3 genes. So anyway, the take home message from this table is to take some liberty that RNA pol means ribosomal RNA. RNA pol 3 tRNA, exclusively tRNA. All tRNA are transcribed by RNA pol 3, means 100%. exclusively. Exclusive means 100%. So all tRNA are transcribed by RNA pol 3. RNA pol 3 also transcribes some ribosomal RNA, okay, and rest all, you can imagine RNA pol 2 take some liberty and make it a little bit simple. All other RNAs are transcribed by RNA pol 2. Keep that in a simple way, that the RNA pol 1 transcribe ribosomal RNA pol 3 transcribe tRNA and some ribosomal RNA, and rest of the all RNAs are transcribed by RNA polymerase 2. Okay, so that's one, keep that one in mind from this table. And now there is subcellular localization of these polymerases. The RNA polymerase transcribed by majority the ribosomal RNA, and the ribosomal RNAs are present in the nucleolus. Inside the nucleus there is a nucleolus, so they are nearby the nucleolus. So the RNA polymerase 1 is also localized nearby the nucleolus. While the RNA polymerase, it is said that in the nucleus, keep that one also in mind, this is the nucleus. Okay, let's suppose this is the cell, and inside the cell this is the nucleus, and here inside the nucleus there is a part that is called, let's suppose, nucleolus. So the ribosomal RNA and ribosomal RNA genes will be here. Ribosomal RNA, this is a cell, this is the nucleus, and inside the nucleus this is nucleolus. So in this nucleolus there will be ribosomal RNAs, okay, and then there will be ribosomal proteins. When these were the ribosomal RNA, so here will be the ribosomal RNA genes also. Ribosomal RNA genes will be basically located nearby the nucleolus. Ribosomal proteins will also be there, and they will be making the ribosomal subunits. They will be forming the ribosomal subunits. They will be assembled, and then they are exported out from the nuclear pore complex. How in the nucleus will look about the nuclear plasm. This is the nucleoplasm. Okay, in the nucleoplasm there is a two zone. One is the central zone, and let's suppose we can say the one is the peripheral zone. So it is said that at the peripheral zone, at the boundary of the nuclear membrane, lies the heterochromatin, so which is untranscribed or be non-transcribed chromatin, or DNA. Okay, and in the center is the euchromatin, or the transcriptionally active active genome. So the most of the transcription happens in the center of the nucleoplasm. So most of the mRNA will be formed here. So the RNA polymerase 2 will be active here, and it will be producing all other RNAs also. So the most important RNA which is produced by the RNA polymerase 2 is the mRNA. This is the most important one, the protein coding genes that produce the mRNA. So in the earlier one, if I say the RNA pol 1 being oversimplified, to keep it oversimplified, ribosomal RNA pol 2 mRNA, and all other RNAs except the tRNA and some ribosomal tRNA and some ribosomal RNA and some ribosomal RNA. So that keep in mind. Okay, and next in this table is given the sensitivity towards the alpha amanitin inhibitor, that is the antibiotic or that is a toxin which inhibit this RNA polymerase. And so this is an alpha amanitin is a toxin as well as it is antibiotic, and by being antibiotics it acts as a toxin for the bacteria. So the alpha amanitin is a toxin, it inhibits the RNA polymerase, and these three RNA polymerases have a different sensitivity. So if I say this is RNA pol 1 and it is given alpha amanitin, so what will happen? No effect on the RNA pol 1. This RNA, so we will call them insensitive, means this RNA pol 1 is insensitive to alpha amanitin. Then RNA pol 2, you get this same alpha amanitin and you give it very low concentration, so it is inhibited. I would say readily or easily inhibited, so we will say it is highly sensitive. So this RNA pol 2 is highly sensitive to the alpha amanitin. And when we will do this RNA pol 3, so we have alpha amanitin, low concentration, so we will see no effect. So what we'll do next, we'll do the same thing but either high concentration. When we will give the high concentration of this alpha amanitin, then we will see the inhibition. Now we will see there was earlier there was no inhibition, but now there will be inhibition. So this RNA polymerase 3 become inactive at the higher concentration of the alpha amanitin, so we will say it is weakly sensitive. So it will be sensitive, it means it will be sensitive at very high concentration of the toxin, a very high concentration of the toxin. And when the concentration is low, generally which is used during the laboratory, there will be no effect. So when there will be no effect, the researcher can conclude that is insensitive, that is insensitive. So keep this one, both the information in your mind, okay, because sometimes the CSIR asks such a questions for which there is no detailed information in the books at the master's level. What they simply say, they simply say, give this graph like this, that the RNA polymerase 1 is insensitive, RNA polymerase 2 is strongly strongly sensitive, and the RNA polymerase 3 is sensitive or inhibited by high concentration of the toxin. So based on this, if we make the sensitivity of the RNA polymerases against the alpha amanitin sensitivity of RNA polymerases, eukaryotic RNA polymerases against alpha amanitin, then it will be RNA pol 2 is highly sensitive, and it is sensitive, more sensitive than RNA polymerase 3, and then the RNA polymerase 1. RNA pol 1 means the RNA pol 1 is the least sensitive, RNA polymerase 2 is the highest sensitive, and the RNA polymerase 3 sensitivity is in between. So that is the decreasing order of the sensitivity of the 3 RNA polymerases. So this should be the information in my mind, but sometime the CSIR asks a question for which it was like confusing options. We will see those examples just now. Okay, so this is the mechanism about the transcription. So we will go back and we will look at some of the questions like this one. Okay, see here, eukaryotic RNA and eukaryotic RNA polymerases and eukaryotic transcription. So this was December 2016 question, and the question number 36 in the booklet A. Which of the following are not transcribed by RNA polymerase 2? Remember, what is not transcribed by RNA polymerase 2 is ribosomal RNA and then tRNA. These two RNAs are not transcribed by RNA polymerase 2, and rest of all of them are transcribed by the RNA polymerase 2. Alright, so which of the following are not transcribed by RNA polymerase 2? So miRNA and some SNO RNA. No, there is no tRNA, ribosomal RNA. miRNA and SNO RNA, no. mRNA and SNO RNA, no. tRNA and ribosomal RNA. Okay, so basically this will be the answer. And if I ask who synthesize, then who transcribe these? Who transcribe these RNAs? These RNAs, the answer should be the RNA pol 3. RNA pol 3. Because the RNA polymerase 3 transcribe the tRNA and some ribosomal RNA, and in the some ribosomal RNA, this is the 5S ribosomal RNA, if I remember correctly. See this one, tRNA and 5S ribosomal RNA. So they are transcribed by the RNA polymerase 3. Alright, so the same question can be asked, you don't know, that simply which or maybe simply which of the following RNA, which of the following combination of RNAs are transcribed by RNA polymerase 3. The same question can be changed by some words like this one, following, then it goes up to here, and then you simply remove this part. Which of the following combination of RNAs are transcribed by RNA polymerase 3? The answer will be the same, the fourth option. Okay. Now if we look at this question, this is the confusing questions, okay, that I was discussing. That in eukaryotes, precursor of the microRNAs and small interfering RNAs are usually synthesized by, no, sorry, this is another question. Okay, we'll come to, are usually synthesized. The eukaryote, the microRNAs and the siRNAs are usually synthesized by, and we know that the siRNA and mi are neither they are ribosomal, neither they are tRNA. Once they are not ribosomal RNA nor tRNA, so they will not be transcribed by the RNA pol 1 or pol 3, so they will be polymerized by RNA pol 2 basically. So the answer will be the RNA pol 2. Now this question, alpha amanitin inhibits only RNA pol 1, RNA pol 2, RNA pol 3 and all the RNA polymerases. Okay, so basically all the RNA polymerases, that is wrong, and we know that this alpha amanitin inhibits the polymerase 2 highly sensitively, and then at a very large concentration it inhibits the polymerase 3 also. So here they have an option, they should have given the RNA polymerase 2 and 3, but they have given one in the option only. So it is a confusion. If I say only RNA polymerase 2, so that will come in our mind, no, that is not the true. The alpha amanitin inhibits RNA polymerase 3 also at a higher concentration. So our mind will say no, oh, what to choose, which one to choose. Okay, so this is the real problem. So always CSIR bring out this kind of questions, so that's create confusion based on the information which the students receive or take during the masters. Okay, from the books, whatever books they are textbooks. So the no textbooks explain in that much of details. So it is only possible when you do some kind of laboratory research. So anyway, since I said at a lower concentration which is generally used in the laboratory conditions, that small concentration of the alpha amanitin will not inhibit the RNA polymerase 3. So we can say, so it will not inhibit the RNA pol 1 because the RNA pol is never inhibited. So the RNA polymerase 3 will also be not inhibited at the laboratory condition concentration. So the answer will be the only RNA polymerase 2. So that will be the answer. So again there is a problem here, but it's the next level problem. But anyway, this question is a little bit better than the earlier one. The alpha amanitin is a fungal toxin which inhibits eukaryotic RNA polymerases. The 3 eukaryotic RNA polymerases show differential sensitivity to these toxins. Which one of the following order, higher to lower, is correct in respect to the sensitivity towards alpha amanitin? And you already know that the RNA polymerase 2 is most sensitive, then RNA polymerase 3, and then the RNA polymerase 1. So the answer will be this one, RNA pol 2 greater than RNA pol 3 and the RNA pol 3 greater than the RNA pol 1. So basically this order also has a problem because it suggests something like that the RNA polymerase 1 also have some sensitivity. Means let's suppose you can say the RNA pol 1 has a sensitivity of 10 and then the RNA pol have a sensitivity of 100, and then maybe the RNA pol 2 has a sensitivity of some 10,000 or maybe whatever. So we'll say this is highly sensitive, this is weakly sensitive, and this is something like that. Okay, but it can be 0 also, it can be 0 also. So imagine it is like a 0, no sensitivity. Okay, so sometime it is also, that order can bring in our mind that all the 3 RNAs are sensitive, all the 3 RNA polymerases are sensitive, but that is not the correct, that is not correct. It should be like a 0 or maybe insensitive. Okay, which of the following statements is generally true about the RNA polymerase 2? It is dedicated to transcribing RNA from a single transcription unit, generally a large transcript, which is then processed to yield 3 types of ribosomal RNA. That's wrong. Okay, because the RNA polymerase 2 has nothing to do with the ribosomal RNA, so that's why it is wrong. It transcribes varieties of small non-coding RNAs which are expressed in all cell types. That could be. It generally synthesizes various types of mRNAs and small non-coding RNAs. Okay, because we know that the ribosomal RNA polymerase 1 means ribosomal RNA, RNA polymerase 2 means mRNA, and RNA polymerase 3 means tRNA in a very simplistic way. So the RNA pol 2 means mRNAs, it has to be mRNA. So that's why the answer is 3. Okay, now we will go back to the mechanism of the transcription. In the case of the, we have already seen it, but we will look at the eukaryotic transcription mechanism. Okay, but we will take a pause here. Okay, and then after a pause, and then we will begin the lecture from this point again.